for joining us today. My name is Susan Evans, and I am honored to welcome you to today's webinar, Care, Cure, Prevent. I have a connection and deep respect with both the neurological team at Stanford and Kensington Senior Living. My father was diagnosed with Alzheimer's disease 10 years ago. Overnight, I went from being a daughter to a caregiver for my father, concerned about the progression of his disease, and my dear mother, who had lost her partner in life. The team at Stanford have supported my father and our entire family throughout this very difficult process. In 2015, we made the decision to move my father to a home. It was the hardest thing I have ever done. The guilt was overwhelming. Caring for my father at home was becoming too much for my mother, and I did not want to lose both my parents to Alzheimer's disease. Our family chose Kensington Senior Living because of their expertise and commitment to dedicated memory care. It was the hardest yet best decision we made. The team at Kensington have taken exceptional care of my father over the last 5 years, and my mother, who pre-COVID had lunch with him daily. I am one of the 30 million caregivers in the country, and I applaud the work that Dr. Longo, Dr. Sha, and Dr. Greicius are doing in the advancement towards a cure. Thank you, Kensington, for supporting and sponsoring their efforts through educational webinars. It is a lonely disease riddled with guilt for the caregivers. Please join me in welcoming Joanne Hubbard, the Executive Director at Kensington Place in Redwood City, where my father is a resident. Thank you, Joanne, for taking exceptional care of my father and also my mother and I. It is brilliant that Kensington and Stanford have partnered together to bring educational webinars in the community. Joanne.
Thank you, Susan. It's a pleasure and a privilege to care for your dad and to partner with you and your family in caring for him and also supporting all of you. Kensington Senior Living is our Kensington Place here is a dedicated memory care. We do have properties across the country from the East Coast to the West, and we are a community that provides excellent care. We have nurses 24 hours a day on site, and we are committed to providing excellence in our care, our clinical outcomes, our engagement, and engaging our family and friends so that each moment is lived to the fullest for our residents. It's important for us to be able to partner with leaders in the industry so that we can help educate the public, so that we can help financially and through awareness to be able to inform people about dementia and to help find a cure. So this is where we have great partners. We partner with Total Care Services to provide great care, but also to sponsor these events with us. And Stanford Neurology has been an incredible benefit to us and a leader in the industry and for our local area here in particular. And so we thank you and we feel honored to be able to do these events with you. Our promise here is to love and care for our residents as if they're our own. And people sometimes ask, well, you're dedicated to dementia. This is what you do. Let it be that we actually find a cure. We can always find other jobs, but that would be an amazing day if we find a cure and there is no dementia to treat. But in the meantime, that's what we do. We're here to provide love and care in the moment for our residents. And we are committed to partnering with Stanford and other leaders in the industry. With that, I'd like to introduce Anne Longo. Anne Longo has been CEO of Pharmatrophix since 2010 and also serves as the Senior Director of Development for Neurology and Neurological Sciences at Stanford University. Following her graduation from Stanford University, she led programs at NIH and at the Alliance for Aging Research in Washington, D.C. Ms. Longo also served in business development at AgeWave and at Charles Schwab in San Francisco. As CEO of Pharmatrophix, Ms. Longo develops and executes funding models, oversees finances working with the CFO, manages commercial projects, and supervises contract research organization projects. She has worked in the medical development since March of 2002, where she served as the Director of Development for Department of Neurology at the University of North Carolina, Chapel Hill, and then worked as the Director of Development at Stanford University for Women's Cancer and the Neurosciences, beginning in May of 2006. In her 18 years of medical development, Ms. Longo has raised over $170 million in new gifts, including 12 endowed medical school professorships. We welcome you, Anne, and thank you so much for partnering with us in this event. Anne?
Thank you so much, Joanne and Kensington, for all the services you provide for our community and beyond. We are so appreciative for the generous support that you have been giving to Alzheimer's and dementia research at Stanford Neurology. Kensington has been a wonderful partner of ours for the past few years, and we are most grateful for this continued support. We at Stanford have a team of over 30 neurologists and neuroscientists working on cutting-edge research to find better ways to prevent, diagnose, and ultimately cure this devastating disease. We provide clinical services to patients and families dealing with all forms of cognitive impairment and dementia through the Stanford Memory Disorders Clinic. In the area of research, Stanford is nationally recognized for its excellence by the National Institutes of Health as a designated Alzheimer's Disease Research Center, better known as ADRC. The ADRC serves as a focal point for Alzheimer's and related research across the Stanford campus and is a part of many national collaborations. We are fortunate today to be joined by three of our esteemed faculty in the Department of Neurology and Neurological Sciences. They have dedicated their careers to caring for Alzheimer's and dementia patients and conducting research in Alzheimer's and other neurodegenerative disorders. Each faculty today will present for approximately 12 minutes, and we will have a Q&A session after the final presentation. Please feel free to submit questions during the presentations through the Q&A button on the bottom of your screen. We'll do our best to address as many of your questions as possible in the allotted time we have after the presentation. I'd first like to introduce Dr. Michael Greicius. Dr. Greicius is an associate professor with tenure in the Department of Neurology and Neurological Sciences at Stanford. He is the medical director of the Stanford Center for Memory Disorders and the principal investigator of the Functional Imaging in Neuropsychiatric Disorders Lab. His research uses imaging and genetics to better understand Alzheimer's disease from the level of molecular pathways to large-scale distributed brain networks and behavior. His lab focuses particularly on the role of the high-risk ApoE4 gene in the development of Alzheimer's disease. By performing next-generation sequencing in protected ApoE4 carriers who show no signs of memory trouble despite carrying this high-risk gene, Dr. Greicius and his team hope to identify rare genetic variants that protect against the effects of ApoE4 and that would offer potential new drug targets. So without further ado, I'd like to welcome Dr. Greicius.
Hi, everybody. I'm going to share my screen. Thank you, Anne, for that kind introduction. Thanks to Susan and the team at Kensington and to Anne for putting this great webinar together. To hide all my beautiful panelists' faces for a second. Okay, so I've got 12 minutes to tell you everything you need to know about genetic risk and early detection of Alzheimer's disease. And right, so we think about the risk of Alzheimer's as divided into genetics and the environmental risks. And we estimate that about 50% of your risk for Alzheimer's disease is genetic. And that means that the rest is environmental. And by environmental, we mean a number of things. It's sort of a broad term. It includes things like age, the hormonal milieu. So for example, whether you're pre or postmenopausal, if you're a woman, your education level. We think education is somewhat protective against Alzheimer's disease. And then a number of other risk factors like head injury, diabetes, and stroke that we think probably also contribute to the risk of developing dementia generally, and maybe also some specific risk for Alzheimer's disease. And an important thing to keep in mind is that your genetic risk goes down as the age of parental onset goes up. And so sometimes, for example, people will come to our clinic and say, I'm really worried about my risk for Alzheimer's disease. Both my parents had it. My father was 102 when he developed it. My mother was 99 when she developed it. And those aren't, that's not a big genetic risk for Alzheimer's disease. That's a big genetic risk for growing very old. And as I mentioned earlier, age is a big risk factor for Alzheimer's disease. So you have to sort of consider how genes and environmental factors interact. All right, when we think about genes, really the one gene that we think about the most is ApoE. This is what's called a Manhattan plot, and it basically shows you a bunch of different genetic mutations or variations across all 22 chromosomes here. We don't include the X chromosome. And basically the higher a point is on this y-axis, the more significant it is in terms of its risk for Alzheimer's disease. And so when you look at this, you might think that ApoE here on the right and BIN1 on the left are similarly weighted in terms of their significance. But the truth is all of these studies have to sort of truncate or shorten the y-axis because the ApoE risk actually goes up to 167 here on this y-axis. So it really dominates the skyline in terms of genetic risk on these sorts of graphs. And it's really the only one that's common enough and causes enough increase in risk that we think about it, day to day in the clinical setting. And so the key things to know about ApoE, it comes in 3 common variations, unfortunately named 2, 3, and 4 instead of 1, 2, and 3. And ApoE4 is the variant that increases risk. So about 10 to 15% of the population, the normal population might have one ApoE4 copy, and about 65% of the Alzheimer's population has at least one ApoE4 copy. So it's much more common in Alzheimer's disease than it is in healthy aging. So ApoE4 increases risk, and ApoE2 is actually protective against Alzheimer's disease. It's also the least common of the 3 variants. People who have the ApoE4 allele and get Alzheimer's disease tend to get it a little bit earlier than most people. So if the normal age of onset might be 70 or 75, people with an ApoE4 gene might present more like at 60 or at 65. And importantly, the E4 effect varies a lot based on your ancestral background. Unfortunately, still for the time being, almost all of our genetic analyses take place in Caucasians. We're working really hard at Stanford and across the country to get more enrollment from underrepresented minorities. But as a really telling example, if the risk for Alzheimer's disease goes up about 4-fold if you have one ApoE4 gene and you're Caucasian, it only goes up about 1.5-fold if you have African ancestry. So it really varies a lot based on ancestral background. We're just starting to uncover some of these differences. The other thing to bear in mind is that the ApoE4 effect is probably stronger in women than in men. So the risk is greater from a single ApoE4 gene than it is in women than it is in men. And that's some important biology that we're still trying to piece apart. And then I think most importantly, in terms of thinking about your genetic risk, we really don't recommend ApoE4 genotyping or genetic testing as a general screening tool. And that's my segue into some really recent exciting advances in diagnostic tests for Alzheimer's disease. And I'm just making the point here that these have gone from increasingly invasive to increasingly less invasive over the last 20 years or so. So it used to be that really the definitive diagnosis of Alzheimer's disease was made at autopsy after our patients had died, looking at the brain under a microscope and identifying some key Alzheimer's signatures. The field moved on and realized that you could obtain spinal fluid using lumbar puncture technique. Just takes about 30 to 40 minutes in the clinic setting. And we can measure proteins that are in the brain that make their way into the spinal fluid. We went from there to PET imaging, kind of like a CAT scan, but with a radioactive tracer that allows us now to see amyloid and tau, the 2 proteins I'll tell you about in a moment in life. And then finally, really incredibly over the last 20 years in my own practice now, getting to the point where blood tests for these proteins are actually hitting the market now and looking quite useful. So you'll never escape an Alzheimer's talk without seeing plaques and tangles. And here's the obligatory plaques and tangles slide. This is autopsy tissue section of a patient who died with Alzheimer's disease. And what you see here in this circle, I'll take it off and put it back on, is what we call an amyloid plaque. This is made up of the protein beta-amyloid, and it occurs in between the brain cells. These are the brain cells here, these skinny little things. So that's an amyloid plaque. And then the other protein that we're interested in Alzheimer's is called tau, T-A-U. And tau occurs inside of neurons. So here's a neuron, and the yellow circle here will show you a collection of tau kind of aggregating, sticking together inside the neuron. So beta-amyloid in plaques and tau in tangles. And so already almost 20 years ago now, some of the first studies started coming out showing us that we could actually measure levels of beta-amyloid and tau in the spinal fluid of living patients. And this is one of the first studies by Sunderland and colleagues, came out in 2003. And what they found is that in a group of healthy older controls, 72 dots here, each dot is a healthy older control, and 131 Alzheimer's patients, the levels of beta-amyloid were much lower in the Alzheimer's group than in the control group. And our way of thinking about this is that the levels are lower in the spinal fluid because all of the amyloid is sort of caught up in the brain in these plaques. And this is a very consistent finding, and it's been seen now, across dozens of studies. So beta-amyloid levels are low in the spinal fluid of Alzheimer's patients. We do lumbar punctures all the time. They're very easy, they're very safe, but they're certainly more invasive than a brain scan. And so just a year after these first spinal fluid studies came out, this incredible study from the University of Pittsburgh came out showing that you could use PET scanning to actually measure amyloid plaque in the brains of living patients. So on the bottom is an old-fashioned PET scan that looks at glucose uptake, sugar uptake. This is a healthy controlled brain looking at their sugar uptake, and this is an Alzheimer's brain where there's less sugar uptake, less brain function in the back. And you can see these are a little bit hard to distinguish on the bottom, but on the top, now we're looking at a PET scan that detects amyloid plaque in the brain. And this is a healthy control with no signal and a patient with Alzheimer's disease with very high, easy to see signal on their amyloid PET scan. So this was an incredible breakthrough. And over the last 15 years or so, this has become more common. It's now FDA approved, although for the most part, not covered by insurance. And then moving still further along the less invasive spectrum, now we have a blood test for amyloid. So a simple blood draw can show us the same sort of things that we saw with PET scans or spinal fluid or 25 years ago in autopsy studies. So here again is spinal fluid in healthy amyloid negative subjects on the left, amyloid positive subjects, those that are likely to have Alzheimer's disease, they have low amyloid levels in the spinal fluid. But what's more incredible now is that we can see very similar changes in these blood tests. So on the left now, I'm showing you the amyloid levels from blood tests in these similar set of patients. So in patients that have a negative amyloid PET scan, they have reasonably high levels of amyloid in the blood. And in those that have a positive amyloid PET scan, we know there's amyloid in the brain, their blood tests show very low levels of amyloid. And so this was very recently FDA approved and can now start to be used in the clinical setting. And I'm not going to walk you through the entire road that tau's gone down, but it's been essentially the same. We started with postmortem autopsy studies, seeing tau in neurons. Then we went to spinal fluid levels of tau. Tau, unlike amyloid, is increased in the spinal fluid of Alzheimer's patients. We moved from spinal fluid analysis now in the last 5 years to tau PET scans. So again, using a radioactive tracer, you sit in a CT scanner essentially, and we measure the amount of tau in your brain. And then most recently, really just this last summer, 2 incredible papers showing that blood tests for tau are essentially almost exactly as accurate as the spinal fluid analyses. So here, sorry about the nomenclature, but these are, CU means cognitively unimpaired. So CU are the normal subjects, and subjects that are amyloid negative and cognitively normal have the lowest tau levels. If you're cognitively normal, but your amyloid test is positive, your tau levels start to go up already. And then these 2 groups both have mild cognitive impairment, but the group on the left is mild cognitive impairment in people that don't have amyloid in their brain. And on the right, mild cognitive impairment in people that do have amyloid, and their tau levels in the blood now are higher than everybody else's. So increasing, it looks like both amyloid blood tests and tau blood tests are actually quite accurate in distinguishing Alzheimer's disease from healthy aging, and importantly, even distinguishing the different stages of Alzheimer's pathology from normal but amyloid positive to mild cognitive impairment and all the way into Alzheimer's dementia. And then in my last slide, I'll just show you how these findings and advances are sort of intersecting. This is data looking at all healthy older subjects. Every time I show this slide, I have to remind myself that nobody in this study had cognitive concerns or abnormal testing. These are all normal older subjects volunteering. And here we're looking at the percentage of these subjects that are amyloid positive on their PET scan. PIB is a specific amyloid tracer. This is the percentage in each age range that are amyloid positive, all healthy controls. And these are split by whether they're ApoE4 carriers in red or non-carriers in black. And so what you can see already in the 60s, age 60 to 69, more than a third of healthy ApoE4 carriers are amyloid positive. And that goes up as we go into our 70s, more than half of healthy, cognitively normal ApoE4 carriers are amyloid positive, and a full 3/4 of them after age 80 are amyloid positive. And you can see in healthy controls, if you don't have an E4 gene, the likelihood that you're amyloid positive is actually pretty low if you remain healthy. So this has really helped us move the field forward, combining genetics with these new diagnostic tests. And as Anne mentioned earlier, my lab focuses particularly on people that have 1 or 2 copies of ApoE4. They're in their 70s or their 80s and they're still cognitively normal. A lot of them will be amyloid positive, so they have amyloid in the brain, but they don't have any cognitive symptoms at this point. We're really interested in studying those individuals and figuring out, do they have other genes that might protect them against ApoE4. So my last slide, I'll just address the recommendations that I make, and this will vary a little bit from one provider to the next. But basically, my general recommendations are, if you don't have any symptoms, you shouldn't get tested. You shouldn't get ApoE genotyping done, ApoE genetic testing. You shouldn't get any of these plasma blood tests. And the reason for that is we don't have any treatments right now, unfortunately, to prevent people from developing the disease. We're working very hard on it. And I think we're going to get there, but we're not there yet. And the lifestyle changes that we would suggest if we find out, for example, that you're ApoE4 positive, are essentially the same lifestyle changes that we recommend for all patients, whether they're at risk for Alzheimer's or not. It's healthy diet, aerobic exercise, and those sorts of things. So it's not going to help us inform you in that sense either. And then if you do have symptoms, I think the first thing you should do is see your doctor and ideally get to a neurologist or another specialist that's got expertise in Alzheimer's disease and related disorders. All right, and I will end there. I don't know how, hopefully close to 12 minutes, 13, sorry. And introduce my colleague, Sharon Sha. So it's a real pleasure to introduce Dr. Sha, a friend and colleague. She's an associate professor here. She is the director of our clinical trials unit. She wears many other hats. She directs our fellowship training program. She has expertise not only in Alzheimer's, but in frontotemporal dementia, Lewy body disease, and in fact is the co-director of our Lewy body Disease Center of Excellence. So with that, I will pass the virtual podium over to Dr. Sha.
Today, I will be talking about our current state of clinical trials. So thank you for that introduction. What I'll first do is talk a little bit about the background of Alzheimer's disease. I think Mike gave a wonderful introduction. The background of clinical trials in Alzheimer's disease. And you may have read about some drug that might get approved by the FDA, by Biogen, and we'll talk a little bit about that. So you already have a bit of background. What is Alzheimer's disease? Just so that we're on the same page. I think we all understand that Alzheimer's disease can involve memory loss. It often can involve word finding problems and difficulty with navigation. And as Mike astutely pointed out, the pathology of Alzheimer's disease is often post-mortem. So like he said, there's not an Alzheimer's doc without pathology. So what you can see on the left here is a plaque that he had shown as well, full of amyloid. And on the right here is a tangle full of the tau protein. And eventually these plaques and tangles cause the damage that we see with those symptoms that we just described. Oftentimes though, what we're seeing, just as he described as well, is that these changes can occur at different times. So it's exciting that we can get blood tests, we can get PET scans, we can get MRIs, we can do spinal fluid. When does that occur in the timeline of clinical symptoms is important for when we intervene. So in this busy slide, I wanted to point out that here on the x-axis is time, And on the y-axis is how we can detect them, what we call a biomarker. So it's a biological marker to detect that underlying pathology. So what really first begins to develop in this purple and red is those changes in that amyloid protein. So he talked a lot about amyloid, when it develops, how it occurs as we age. So that's the first thing that changes over time. Then we can start to detect changes in tau in this light blue in the spinal fluid. Where tau PET and where tau in the blood can be detected has yet to be determined. Then we can start to see changes on the MRI. So the MRI, you can see atrophy, so brain shrinkage. We can see hypometabolism, so decreased metabolism on a PET scan, a glucose PET scan later in time. And then finally, we start to see these clinical changes where someone can start to have memory problems, word finding problems, and eventually the diagnosis of dementia because those cognitive problems cause now decline in independence. So from the onset of the changes in the amyloid that we can detect to a diagnosis of Alzheimer's disease where someone has dementia, now this functional decline, that can be 15 to 20 years. Hence, most of the clinical trials have been targeting this amyloid protein because it's the first thing that changes. If we can prevent that from happening, a lot of people in the field have started to wonder, maybe if we prevent amyloid buildup, could all of the downstream processes, everything that occurs afterward, could we prevent that? And that's been the nature and the thrust of why a lot of clinical trials have targeted amyloid. As you may know, we have no disease modifying drugs. And what does that mean? Essentially, over time, we know that memory declines. So that's what this is representing. And then what we have now is symptomatic therapies for Alzheimer's disease. So you may have heard of donepezil or ribostigmine. Those medications are symptomatic, meaning they delay the symptoms. So that memory loss still occurs, but it just delays it. What we're really looking for is something disease modifying. So it changes the slope, maybe even improves memory. And as I said earlier, a lot of the drug targets have been focusing on the amyloid protein. So what we have in this cartoon is this amyloid here. So what happens is this amyloid precursor protein gets cut by a bunch of enzymes, alpha secretase, beta secretase, gamma secretase. And some people have thought about preventing that cleavage process, preventing that cutting, and then preventing that amyloid from building up in the first place. Another way is to get rid of the amyloid. So if we can get something that binds to it and gets rid of it, what we call these amyloid antibodies, that's a potential method of reducing the amyloid. And there have been several amyloid antibody trials that have been ongoing. Essentially, they have all failed, though. So as you can see here, there have been at least 6. One is ongoing, and we're going to talk about aducanumab in the later slides. And one is recruiting. So that's ongoing right now, and I wanted to tell you a little bit about this trial. So the company Eisai and Biogen created a drug called BAN2401. And in an early phase trial, so there's several phases to trials, phase 1 and phase 2, phase 3 and phase 4. The early phase trials has a lot of a smaller number of patients and really is looking for safety in the patients. And when we get to phase 3 trials is when we're really looking to see if the drug does what we think it does and really changes that disease course. In this diagram, what we have is over time, again, what the study looked at was about 800 patients who got either placebo, so that's the fake drug, or different doses of the drug, and see if it changes a composite measure, so a combination measure of independence and memory. And what they found was that over time in the placebo, where people get worse, with the drug, they got less worse. So that was exciting. The problem with this clinical trial was that they really converted a lot of the ApoE4 carriers. So what Mike had just mentioned, patients who have that particular genetic mutation, they removed them from that treatment group and put them in the placebo group. So the problem with that is that when we're really trying to see if a clinical trial is working, you want the two groups to be the same. If there's any difference, like if you only have men in one group and women in the other group, we don't know if that can be applied to the other group. So now it's going through a phase 3 trial to really see if this, what we found, that change works. So we're excited about it, but very tempered just because of the problem, we can pause here, the problem with actually knowing whether or not it will be successful. But we're very excited about it, and we are recruiting for that, and we're running that clinical trial at Stanford. Now we wanted to talk about this aducanumab one by Biogen. If you could advance the slide, thank you. An early study of only 166 patients demonstrated that maybe there's less progression over time. So what you're seeing in this picture here is very, very similar to what Mike showed of those amyloid PET scans. On the left is what you see is what when someone who has Alzheimer's disease and they have amyloid in the brain, it looks red. When on the right column, we see patients who got the drug, and people who had the highest dose of the drug had removal of that amyloid. So you can see now yellow instead of red. So that red hot was removed. And that's very exciting. It demonstrates what we say target engagement, meaning that the drug gets to the target and we remove it. But who cares if we remove amyloid if it doesn't affect memory? Can we get to the next slide? So what they showed in this study is that in a dose-dependent fashion, almost, meaning the highest dose, there was less decline in memory. So this MMSE is a little brief test out of 30 points. And we know that the placebo, which is yellow, people get worse over time, about 3 points per year. And so that down is the decrease. But what you see in the blue, the dark blue, is on the highest dose, they had less of a decline after 1 year. So that was very exciting. But who cares if you remove amyloid and the memory gets less worse if you're still functionally less independent, that you can't be independent as much? Can we go to the next slide? So what they showed on what we call the CDR sum of boxes, that's the clinical dementia rating scale. And it's a score basically to see how independent someone is. Again, in the yellow, we see people get worse over time. That's to be expected. And then the high dose, that dark blue again, people got less worse over time at about a year. So that was very exciting, but it was only 166 patients. So that was a phase 1B trial, so an early phase trial, really just looking for safety. So then they went into a phase 3 trial. Next slide. And so they recruited thousands of patients around the world. And then in March of last year, they canceled the trial. All of a sudden, it was all over the press. Even some patients heard about it in the press before they heard it from their doctor. And it was very sad. Essentially, a different advisory group had analyzed the data that they had collected thus far and said that it was going to be futile to continue. They weren't going to meet their primary endpoint, which is that CDR sum of boxes, that functional independence. Next slide. But then in October of 2019, there was a switch, this flip, whereby we heard from Biogen that they said that they were going to file for approval for the drug. Why would that occur when they just canceled it in March? Next slide. And so a lot of people wondered what was going on. Next slide. And now what had happened was that they collected more data after they canceled the study in March and said, actually, when we reanalyze the data, we think it might be successful. So we're going to file with the FDA and try to get this drug approved. Just recently, you see, this is from Time Magazine on November 10th, the FDA had an advisory committee and said, let's look at this data. And the advisory committee thought the data was not appropriate to get approved by the FDA. The FDA now must decide what to do with this information. Next slide. And let me break down the data as far as we know. So there were 2 concurrent studies in parallel that essentially recruited the same number of patients. And they really got 2 different doses of the drug. And what you can see in the red is that over time, there is no change in this amyloid PET. So this is really looking to see the amyloid was removed over time. And in the placebo group, red, no change. That's to be expected. In the blue and the green is the high dose, people had reduction in the amyloid PET, which is great. That's what we saw in the early study. So we think that there is what we call target engagement. But again, what do we know about memory and what do we know about independence. Next slide, please. And that's where the data diverges. So it's really confusing as to why one study would be successful and another study was not successful. So with the eMERGE study, if you look at the top column on the left, the CDR sum of boxes is what the primary endpoint is, meaning that that needs to be successful for us to say that this drug is working. And that's a measure of independence. If you could go to the next slide, what we're really looking at is to see if it's statistically significant compared to the placebo. And what we find is that they're different. That usually significance is defined as the p-value less than 0.05. And we have that for the eMERGE study. But the ENGAGE study that had the same number of patients was not statistically significant. We find that on measures of cognition, that ADAS-Cog 13, they did find that there was improvement in memory, or actually less of a decline. But ENGAGE, they didn't. And then another measure of independence, the ADCS-ADL, they found that there is less of a decline in the eMERGE, but not the ENGAGE. And how do we reconcile these two diverging studies? One was positive, one was negative. Next slide, please. And it's not without side effects. So here's the issue. I think if we said, well, maybe this drug could be doing something, maybe it's worth a try. The problem is the way that amyloid antibodies work, sometimes it causes brain swelling. So they call it ARIA-E. ARIA stands for amyloid-related imaging abnormality. That's a mouthful, so they call it ARIA. E stands for edema. So what you see on that left panel in A is that there is some bright white, and that is that swelling or edema. There's about 35% of patients who got the drug had this ARIA-E. A majority of them were asymptomatic though. They had no symptoms, and we really captured them when they were in the trial by getting repeated MRIs. A smaller number of them had ARIA-H, which stands for hemorrhage. So those black dots in B are little microbleeds. And about 20% of the patients who got the drug had a headache. So there are some side effects. So it's not without caution that we think, could this drug get approved? Next slide. So that's that's almost my final slide to say it's a huge conundrum, and you can tell that in the field of experts we have a lot of people on different sides, especially if you believe this amyloid hypothesis that targeting amyloid and getting rid of it can fix everything. The problem is when you have two studies that are different in their results, what do they mean? Does this drug do what we think it does? Or maybe they should actually finish the study instead of canceling it out early. And with that, next slide. I wanted to just say thank you for your patience and attention as the slides kept crashing on me. Okay, we can stop sharing. I wanted to take the time to introduce our next speaker, Dr. Frank Longo. I'm very excited to present to him. He's been a great mentor and fantastic chair. So he started his residency at University of California, San Francisco. He also completed his fellowship there in neurobiology. He stayed on as faculty and eventually served as professor and vice chair of the Department of Neurology. He joined Stanford in 2006, where he now serves as our illustrious chair. He is focusing as chair on building programs in neurology and neurosciences. His interests include translational research in neurodegenerative diseases and therapeutics. And he sees patients in the memory disorders clinic with myself and Dr. Greicius. And he also mentors our medical students and other trainees. He's a fantastic teacher. His accolades are including being the 2015 recipient of the inaugural Melvin R. Goods Prize for Excellence in Alzheimer's Drug Discovery from the Alzheimer's Drug Discovery Foundation. And his work in Alzheimer's therapeutics was featured on the cover of Time magazine in 2016. It's a beautiful picture with some really cool people. Thank you.
Well, thank you so much, Sharon, for your introduction there. And thank you everyone for joining us. Also wanted to thank Susan Evans for helping put the program together today, and to Joanne Hubbard and her team at Kensington for being great partners for us here at Stanford and the great care that you give your residents there. I'm going to go ahead and share my screen here and share a few slides. And we're going to talk about what it's like to develop a new therapy and share our adventure in developing a new therapy for Alzheimer's. Okay, so we'll talk about what it's like to develop a new therapy. And part of the problem with Alzheimer's that I'd like to highlight, Dr. Greicius and Dr. Sha shared how in our patients with Alzheimer's, we have amyloid and another abnormal protein called tau accumulating in the brain. And the reason why those two substances are so toxic is because they interfere with the connections in the brain. So I'm sharing with you here a picture of a neuron, the cell in the brain that allows it to function. And it's all about the connections. So with these blue arrows, I'm showing some axons, and they're coming in and connecting with the dendrite of this neuron. And along the dendrite there, you see these little outpouchings. Those are called spines or dendritic spines. And those are delicate structures there, but they're the very heart of the synaptic connection. So this red circle in the middle is showing you synaptic connection between the incoming axon and where it connects to that next neuron, and the electrical signals going across that synapse, that connection. And that spine, those little nubs are the most delicate part. And the reason why I'm highlighting those is because they're so delicate that they decrease, the number of those decrease as we age, but they especially decrease in Alzheimer's disease. And the bottom line of why our patients are losing their memory and other function is because they're losing these connections and they're losing those spines. So we need to find a treatment that prevents the loss of these spines, prevents the loss of these connections. And so Dr. Sha shared with you going after amyloid as a therapy, and it makes sense. We think amyloid triggers much of this. However, the problem is that very quickly, possibly after the brain starts accumulating amyloid, that triggers the accumulation of this abnormal tau, but not only amyloid and tau, we start to get inflammation in the brain. I'm showing a picture here of a microglia cell. That's the brain's equivalent of an immune cell, like a white blood cell. They're activated in a toxic way in brain inflammation. We see that occurring in Alzheimer's disease. And finally, with aging itself, we could see neurons and those dendritic connections begin to shrink just from aging. So we may have all four of these processes contributing to the loss of these spines and synapses in our Alzheimer's patients. And maybe these treatments that are going after amyloid only, unfortunately, may not be all that effective because we still have these other processes going on. So our dream is to develop a drug or a treatment that can go after all four of these degenerative processes all at the same time, and perhaps be even more powerful than just going after amyloid. So again, we're coming back to these spines here, and I want to show you something that's called resilience. And this is one of the most exciting things in the field, I think now, this concept of resilience. So this is a normal human brain showing the dendrite that we need for the connections, and those little nubs there are the spines. And you see the spines coming off this normal dendrite in the normal human. Now in the person with Alzheimer's, it's accumulated the amyloid and they have dementia. You see the loss of many of those spines. We don't have as many of those spines, those little nubs coming off here, and they're losing their memory, right? But in some people, Dr. Greicius mentioned this, it's a big mystery. We have some people who have just as much amyloid accumulated in their brain. They have just as much aging. However, they have normal cognition. In other words, they're resilient. And when we look inside their brains, trying to understand the secret of why they're resilient, look what we find, a normal number of spines. Those spines and synaptic connections are holding up to the amyloid, and their cognition is normal. So could we replicate that with a therapy? Could we make those spines and connections resilient to amyloid with a therapy? Well, here's our experiment in mice. We've got a normal mouse with plenty of those spines. This is a mouse with a normal memory. But then we can also have mice with Alzheimer's disease. We put human genes in these mice that run in Alzheimer's families. And you can see this Alzheimer's mice has this lower number of these spines. And this mouse also has memory problems. We can test the memory of these mice. However, when we give those Alzheimer's mice one of our drugs that we've developed in our laboratory, look what happens. We have a normal number of spines. The spines come back, and in these mice, we have not lowered the amount of amyloid. In other words, we've invented a drug here that makes the brain resilient to amyloid and makes the memory normal in those mice. Can we do this in a human? That's the goal. And so we do many tests on our mice that mimic what we do with humans. Here's an example of a PET scan. And our mice, we treat them very gently here. They're sedated and they're getting their PET scan. And we look at the brain down here at the bottom. The normal mouse has this bluish color. That's a normal brain. But in the Alzheimer mouse, this greenish color showing us signs of inflammation in the brain. But look at the mouse that was given our drug. That green signal for inflammation is almost entirely gone. So we start with the mice and we test the mouse in a similar way that we test people. Let's move into the people now. This is not easy. On the left-hand side, this is called the drug development pipeline, the therapy pipeline. And on the left-hand side here, I'm showing you the work we've done in the university lab that I share with you. And our goal, of course, is to get to these big phase 3 trials that the large pharma would want to do. The problem is those big phase 3 trials are very expensive, very risky. And so our big pharma colleagues be very careful about what drugs they end up testing, right? They can't have too many failures. So you have this in-between zone where we can get it so far in a mouse in the university. Big pharma can't take these too big of a risk. So the middle zone is sometimes called the valley of death. How do you get a treatment through this middle zone so that a big company could take up the drug and do the big phase 3 trial? And the one way to cross this so-called valley of death is to start your own company, to do these basic studies of safety in rats and dogs and your early trials to see if you're safe in people, and what we call a phase 2 early trial in Alzheimer's to see how the drug works so that it can go on to the big trials. And so that's what we did. We started a company called Pharmatropics, as you heard. Anne Longo is the CEO and made that company possible and figured out how to finance that company. And to make a long story short, through that means we ended up being able to do a phase 1 safety trial with one of the drugs we invented. And this is a big deal. This is a new drug. There's never been a drug like this in a human being before. And the FDA requires that it goes into normal people for the very first time. So there are fortunately normal, healthy people that will volunteer to take this drug for the very first time. Here's a testing center. Doctors and nurses are there 24/7 as the very first people take this drug. This is the control room. And fortunately, this drug that we invented is safe in normal people. That was extensive testing. Once we found out it was safe in normal people, we were allowed to move forward and test this in Alzheimer's subjects. And we've just now completing what we call a phase 2 exploratory endpoint randomized controlled trial in 5 countries in Europe. We were able to move a little faster, a little less expensive in Europe, and the patients there are a little bit more tolerant of getting spinal taps, so that makes the study more efficient. So we enrolled about 240 Alzheimer's subjects. 80 were on placebo, 80 low dose, 80 high dose, all blinded, treated for 6 months. And this is for safety, and then we're looking to see if we have any effects, good effects on the brain. We don't have the answers yet, but we hope to have some initial results by the end of this year and early 2021. It's been quite a journey, inventing a new treatment, getting it through the mice, getting it through the safety, getting it through normal people. And this is us on family vacation, visiting clinics throughout Europe. Rather than visiting the Eiffel Tower, we went to neurology clinics. And this is Anne and I at a clinic in Barcelona. And here's a patient just returned their empty capsules. This is a capsule that's taken orally twice a day. And it's really inspiring to see that. And because this journey is so unusual of inventing a new drug and bringing it through all these phases, the story itself ended up on the cover of Time magazine, as Dr. Sha mentioned. Of course, it'll be even more exciting if it's effective, right? That's what really counts. Fortunately, this is a capsule, as you can see. These amyloid treatments that Dr. Sha described are intravenous treatments that will be required once a month, probably. They'll be very expensive, but our drug will be what we call oral, not intravenous, very low cost, and anybody in the world could take this. And as you know, we're approaching 50 million people in the world dealing with this. Our dream is to have a low-cost drug that everyone in the world can take. So I will wrap up there and leave time for questions.
And Dr. Longo, I will just quickly take a moment here, if I may, I'd like to just share one slide. Can you all see that?
Yeah.
Great. So I'd like to just invite any of our attendees today to join Kensington. Kensington has been very generous over the years in helping to support our research programs. So if you're interested in supporting and advancing Alzheimer's and dementia research at Stanford, the information is all here. And you can contact me anytime. Email is always best with any questions around fundraising or clinical issues, research issues. Please feel free to contact me. If you'd like to make a donation, the easiest ways to do so would be via check or online with a credit card. So if you'd like to send a check, just make it payable to Stanford University and make sure you write in the memo line of the check, Alzheimer's Fund GHFRP, and mail it to this address. Online via credit card, visit makeagift.stanford.edu and select the Stanford Medicine and then type in the same fund number in the special instructions section. And right now, I would like to invite the speakers to come back for our Q&A session. We have about 5 minutes. We don't have a whole lot of time. And we have a fair amount of questions, and I'm going to just pose maybe 3 or 4 because we're limited on time right now that really would be broadly applicable to the entire audience today. So then some of these questions I might direct specifically to one of our faculty versus leave it open. First question I'd actually like to direct to Dr. Sha. Dr. Sha, is Stanford currently running clinical trials during this pandemic?
Yes, thank you for asking that. You know, obviously in the beginning of the pandemic, we were very cautious. A lot of our studies were on hold because of our vulnerable populations. And given the changes and more knowledge about the coronavirus, we've implemented a lot of safety measures, and we've started to open up a lot of our clinical trials. And research must go forward. You know, obviously, as you've seen today, the research is really important to finding a cure. So if I may share my slides with the risk of crashing yet again, I only have 3, but essentially the current trials that we have right now that we're currently recruiting for is one, the BAN2401 that I talked about, where we need to find out if it truly does slow down the disease progression. And another one for moderate to advanced Alzheimer's disease and that causes agitation. So in Alzheimer's disease, we have agitation. There's one potential treatment. We have a number that are ongoing that are not recruiting. One is upcoming for prevention. So someone who's cognitively normal, but at risk for developing Alzheimer's disease because of amyloid presence in the brain, we may be giving the same drug, BAN2401. There are a bunch of observational studies that are recruiting. Our ADRC, Alzheimer's Disease Research Center, recruits a bunch of different kinds of participants, including cognitively normal, Alzheimer's disease, mild cognitive impairment, Parkinson's disease, Lewy body disease, and soon we'll be recruiting early onset in a specific clinical trial or clinical observational study called LEADS for longitudinal early onset Alzheimer's disease. That PSAD study is basically the results from a pilot study of a drug already approved called prazosin. It's a drug that's for blood pressure, but in the small number of patients that were given prazosin, that improved agitation. It was well tolerated. So now we're moving forward with a clinical trial to see if that works. Essentially, the prazosin helped agitation, whereas the placebo didn't help it as much. So what I really want to share with you is to make sure you get involved. You know, whether you make a gift to us, support research, it's really important so that we can learn from this and then share this back with you all again. So these are the numbers and contact information for our various clinical trials. And please get involved. Thanks.
Great. Thanks so much, Dr. Sha. Here's another question that would apply to everybody. What is the latest on exercise? Is it still showing to help prevent or slow down the progression of Alzheimer's or dementia?
I'll answer that. The exercise studies continue to look good. More and more studies are done, and they slow down the accumulation of amyloid in some studies. And I don't think it'll be a cure-all, but I think for any of us at risk, I think the exercise could potentially delay onset or make the disease go slower. So it's worth it. Studies still look good.
Great, thank you. Another category that broadly affects everyone, what about sleep? How does sleep affect our risk for progression of Alzheimer's dementia?
Maybe Dr. Greicius could take that.
Yeah, I'm a little bit of a skeptic. I mean, sleep is obviously very important for memory, and I think everybody has experienced having a crummy night's sleep and doing worse the next day in terms of your cognitive function. You know, there's a lot of concern that maybe sleep apnea is increasing risk for Alzheimer's disease. That most of that data still, and the sleep data in general, come from animal models. We are getting more and more human data on this. It's hard to do, but for example, doing sleep studies while also measuring levels of amyloid and tau. And it does seem as though a good night's sleep in deep, slow wave sleep is important for clearing amyloid, for example, from the brain. So, you know, I think it's a very important and open question, basically.
Great. And then in the interest of time, I'll ask one last question. This is posed to Dr. Longo. When is your drug going to prime time? When will it be ready? Say if you do get positive results from your phase 2 trial, how long would this take to get to market for patients?
Yeah, if we get positive results on the phase 2 trial, that we hope lead to a phase 3 trial, and a phase 3 trial can take 3 or 4 years. So we're at least 4 or 5 years away from this being available. And I'm sorry it's so long, but that's how long it takes.
Great. And again, we're sorry we cannot get to all your questions. I'm sure we'll have another session in the future and we can address more of your questions.
We'll have another session in the future, Anne. Thank you so much. I can't thank Frank, Sharon, and Mike enough. Valuable information. And, you know, the numbers are staggering with 50 million people with Alzheimer's and an average of five people as caregivers for those people. That's, you know, 250 million people in the world as caregivers. And really my heart goes out to them, especially during COVID, during this difficult time. I want to let everyone know that joined us today that you will be receiving a copy of the webinar via email. And I also encourage everyone to support the work that Stanford Neurology is doing. We need to find a cure, and all of your help can help move this needle forward towards a cure. And the work that you're doing is so incredibly important and dear to my heart, especially. And so Giving Tuesday is coming up, and so please consider joining Kensington Senior Living and making a donation to Stanford. Joanne?
Thank you, Susan. Thank you so much, everybody. We so appreciate all of your work here and all the work that you're doing to find a cure for Alzheimer's. And we would like to present you with a $10,000 check for Stanford University for your research. So thank you so much.
Thank you.
Thank you so much. Thank you so much, Joanne and Kensington. That will make a big difference and accelerate what we're doing. And we enjoy partnering with your company and the great care that you're giving your patients and families.
Thank you so much.
Be safe and have a good Thanksgiving.